Hello, welcome to today's coffee seminar. It's my pleasure to introduce Professor Huang from the University of California, San Francisco. Dr. Huang received his bachelor degree in chemistry from Peking or Beijing University in China, and his PhD degree in chemistry at Stanford. He worked as a postdoc at Harvard University and then joined the Department of Pharmaceutical Chemistry, University of California, San Francisco. His research work encompasses the area of single molecule biophysics, optical microscopy, and bioanalysis. And he has received many awards, including the GE Healthcare and Science Prize for Young Life Scientists, the Searle Scholarship, and the NIT Director's New Innovation Award.
Well, thank you, and thanks for, well, I'm really honored to be invited to come here. It's my first time to Boulder, and it's really beautiful campus and great people. Okay, here, just to make everyone jealous. This is seawater, San Francisco Bay, and it's the view from my window of my office last week. And you see here, this you get after a storm. And so that comes to the main technique that I want to talk about is storm microscopy method with how we can make it faster, higher, and stronger. That's following the Summer Olympics. So a little bit of background on the scale that we're trying to study. And of course, all these methods is trying to understand how we are here, how we get old, how we grow up, how we get sick, how we can get back healthy again. So we want to look at some human beings. This is at the meter scale. And if we want to understand, for example, how the brain functions, then we'll have imaging methods. That's looking at the brain. This is like decimeter scale, and of course, for this particular person, the brain anatomy is definitely not like this. It's like this, and even based on that, probably we can define the new species other than Homo sapiens as Homo simpson. And even for a simple brain like that, it's still made of a very complicated neuron networks of neurons. And they talk together, they get our brain working, and that's at the sub-millimeter, millimeter, centimeter scale. And all these scales are very much covered by CT, MRI, those like in vivo whole body imaging techniques that can look at both human and animal. But if you want to look deeper, for example, and find out how a neuron works, and that's belongs to the scale of optical microscopy and going millimeter down to micron scale. If you want to go further deeper, like I'm a chemist, I'm trained chemist, so I'm interested in molecules. I always want to understand how a molecule works. To a molecule, even the cell is going to be a monster at micrometer, multiple micrometer scale. The big protein complex, for example, this is Nobel Prize winning complex, the ribosome is 100 nanometer. In 70 nanometer, so this is the scale of protein complex. A protein molecule is going to be several nanometer. This is a Nobel Prize winning molecule, GFP, although it's not the most fashionable one. Now we should all talk about G-protein coupled receptors, because this is this year's Nobel Prize. And if we go down to a small molecule, we have to use Angstrom resolution techniques to look at the structure of these molecules. So night microscopy, we want to look at, it's very easy to look at cells. But it's not so powerful looking at things smaller than a cell, because the diffraction of light, the physics limits the resolution to about 250 nanometer, 300 nanometer. That's about exactly the scale of organelles inside a cell. So there's no hope for protein complexes. And for these things, we have an array of structural biology methods, like crystallography, like NMR, like single particle cryo-EM, to look at these proteins, protein complexes, and we know its atomic structure. But if you look at this thing, there's a gap of a resolution between cell biology methods, especially light microscopy and structural biology methods. And then that creates the problem of how we can link our understandings of molecular scale function versus molecular scale structure to cellular scale function. And that is the void where super resolution microscopy, particularly STORM, PALM, STAT, structural dimension microscopy, all these techniques coming in to fill in. The method that I'm going to talk about is STORM microscopy. I'm just going to go briefly about its basic principle. And so we have a structure in a cell, say these 3 lines. And we know, well, any of the structures are made of discrete molecules. We never have continuous structure in the cell. And supposedly, all these molecules are fluorescent because we capture its fluorescent image. Because of the diffraction of light, we end up with very blurry image, not able to resolve them. So this is the case with all molecules are fluorescent. They emit fluorescence, their images all overlap. So one way that would allow us to distinguish molecules next to each other is to have molecules in different chemical states. So if 2 molecules next to each other, one is giving out a fluorescent signal, the other one is not, then we can definitely say, okay, all this signal comes from that molecule. And then we can distinguish the 2. And this is the key for STORM and PALM. And because once you can isolate single molecules, you can capture single molecule images. So this, all these photons are from one molecule. And that will allow you to do a lot of things. For example, fit the shape and get the position of that molecule. And this modulation is enabled by a class of fluorophores. They call the photoswitchable dyes and photoswitchable fluorescent proteins. And they can be in 2 chemical structures. One is absorbs light and gives out fluorescence, and the other one does not. So what you do is you have all these different molecules in the sample, and you put them all in the dark state, and then you use light to convert a small fraction of them into the fluorescent state. And because it's such a small fraction, you got a very sparse subset of these molecules that's being switched to the fluorescent state. You can capture the image, for example, then you can determine their positions, and you switch them off, for example, by photobleaching them. And then give the activation light again, and that will switch another subset of these molecules. So you can determine the position, you let this process go, and you can start to see accumulation of localization points. And that will allow eventually reconstruct the image by knowing the positions of all the molecules in the sample. The switching process itself is stochastic. We don't control which one to turn on, which one to turn off. And so that comes the name of stochastic optical reconstruction microscopy. And also this process involves photoactivation and localization. And that comes the name of photoactivated localization microscopy, or PALM, and fluorescence photoactivation localization microscopy, FPALM. The 3 methods essentially the same thing. It comes from Xiaowei Zhuang's lab at Harvard, and Harold Hess and Eric Bettig and Geneva Pham and Sam Hess at University of Maine independently in 2006. So there's 3 different names for one method. So that's the basic principle, and we're going to come back to that again. One example of how we can use it. This is microtubules in a cell. We do immunofluorescence using antibodies to stain these microtubules. And the antibody is labeled with a dye called ALXS647. And it's a commercial antibody, but under certain chemical condition, it's photoswitchable. So we can do the photoswitching and determine the positions, reconstruct an image. And this is reconstructed from a total of 40,000 frame raw data, 1.5 million single molecule localization points. And from the zoomed in image, we can see the resolution improvement to allow us to resolve individual microtubules. And this method can also be extended to 3-dimensional localization. This is some work that I've done when I was a postdoc in Xiaowei's lab, and is to introduce some optical aberration in the imaging path. And for example, here, when we scan the stage from below the focal plane to above the focal plane, the cylindrical lens would cause the shape change of these molecules. Then we can use the shape to determine the Z position as well as the XY position from the center. So that will allow us to reconstruct a 3D image. And this is just one of the methods that can allow us to determine 3D position. There are other methods like biplane imaging, like the double helical or all the other points of function engineering effort that Rafael has been working on. But so they all come to give us 3D images of serial structure. And this is an example taking of my collaboration with our collaboration with the Pietro Di Calimini lab at Yale University, imaging clathrin-coated pit. These are in vitro reconstructed clathrin-coated pit. So it's a pit. That means a hemispherical cage structure with an opening. We can image this protein structure. You can see it's round shape. The diameter is about 150 nanometer. That's in fact smaller than our camera pixel. But we can determine the 3D position, do a 3D reconstruction. So if we take a cross sections, you can see this empty shape, or if you take a cross section through the rim, you can see the opening or the top is closed. So we can resolve this structure completely. And overall, we got a precision of about 20 to 30 nanometer in determining the XY position of the molecule, and about 50 to 60 nanometer to determine the Z position of that molecule. And so in full with half maximum. So in all three dimensions, it's more than an order of magnitude improvement over either confocal or two-photon microscopy. With this resolution, 20 to 30 nanometer, we can come back to our resolution plot. 20 to 30 nanometer, in fact, it's already smaller than many protein complexes. It's getting to the size of the protein molecule. So that's why I say superresolution microscopy serves as a method of the tool that can bridge this resolution gap between structural biology and cell biology. We've used this method to image many different structures, like 3D imaging of microtubules. Now microtubule images being too commonplace. So we're adding mitochondria in addition to microtubules to the two-color 3D imaging, and or clathrin-coated pits. So this is our collaboration with the Calimini lab, showing that the membrane tubule underneath the clathrin-coated pit being coated with a protein FP17. And we have also collaborated with Catherine Dulac lab at Harvard, looking into mouse brain sections. And this is looking at one synapse of between two neurons. So you can see the oval shape of the active zone as well as the synaptic cleft between the pre and post-synaptic site. And you can see the size of a synapse is in fact smaller than the wavelength of light. And one specific example, and this is our recent collaboration with David Agar lab at UCSF, trying to look at a structure inside a cell called the centrosome. And from the name, this is a structure that's locating at the center of the cell. And it has two very mysterious perpendicular cylindrical shape of centriole. And outside the centriole, there's a so-called amorphous structure called the pericentriolar matrix. And the pericentriolar matrix is a linkage between the centrosome to the microtubules extending out from the centrosome. So it organizes microtubules so that it stays in the center, so that the cell know where the center of the cell is by following the microtubules. And people have done extensive electron microscopy studies of the centrosome. From these electron micrographs, you can see the centriole, but not quite about the pericentriolar matrix because it's made up of proteins. And electron microscope cannot tell, okay, this dark region is protein A, this dark region is protein B. It just sees some protein dense structure. And that's why we call it a matrix. So for the pericentriolar matrix, it's really like a spaghetti ball. And this is a postdoc, Vito Manena, in David's labs trying to look into using superresolution microscopy. And the paper has just been published a week ago online in nature cell biology. And in this case, the central zone structure is about 200, 300 nanometer. So there's no hope no hope to resolve it in wide field fluorescence microscopy. Video's been mostly using structural illumination microscopy, even by just the factor 2 improvement in resolution. So structural illumination microscopy has a resolution about 100 nanometer, and that you can see substructures within the central zone. And to make a very long story short, we focus on one of the proteins. It's called pericentric-like protein, PLP. And this is a very long protein. The known isoform has about 3,000 amino acids. And Video has used antibodies that detect 3 different domains on the same protein molecule. From this image, because we are looking at something surrounding the centriole, you can clearly already see the ring diameter is different. To be more precise, we take a radial cross-section and measure radial distribution. The reference mark we have here, SAS4 marks the centriole. This is the wall of the centriole. Gamma tubulin marks the microtubule organization center. So that's where the microtubules start to grow out from the central zone. And the C-terminal packed domain located exactly at the centriole. The mid domain is 30 nanometer extending outside. And the N-terminal domain is yet another 30 nanometer towards the outside. So from this model, we know that this protein has a highly oriented structure, going from the centriole extending out. If we do higher resolution of storm, we can see it's not quite a ring, but discrete clusters surrounding the centriole. So overall, the model will be that the pericentric-like protein, PLP, has the packed domain binding to the central wall, and the C-terminal domain and N-terminal domain extending out. We have biological experiments showing that if we kill this protein, the entire pericentrial matrix will collapse. So this is almost like the stem of the tree of the PCM. So it's not quite like a spaghetti ball. Looks the same from a far distance, but it has some order structure in there. So this is the one example showing the application of superficial microscopy in understanding a biological structure and also in helping to understand its function. And what are we really going to do? One thing we really want to do, if we could push the resolution just by another factor of 2 or a factor of 4, then we're getting to the size of the protein. So higher resolution, then hopefully we can resolve protein structures, really individual proteins. And that has been a series of efforts of looking for bright fluorophores. Some of you might recognize, I don't know if any one of you might recognize this fluorophore, hydrocyanin. New fluorophores supposed coming up soon from Shelley Snap. Also trying to find better neighboring methods. Some of these neighboring methods we are working on, but we're also taking the effort of, we're also trying to learn from the efforts of other groups, for example, using DNA and RNA aptamers as well as the nanobodies to enable cellular structures. One of our own method is to improve the nucleation sites and using the mirror to reflect the other sides of the fluorescence going to the other side back into the objective and using the spatial light modulator to create interference between these two fluorescence emission paths so that to allow us to get better nucleation precision in a Z direction. So all these efforts we've been trying to get better spatial resolution. And because there's a lot of chemistry and biochemistry effort here, because we don't have time to go into many of detail, but if you have any questions, I'll be happy to answer questions about this after the talk. One effort by going into higher resolution is to resolve the structure of the nuclear pore complex, which is a pore in the on the envelope of the cell nucleus that controls, for example, transcription factors getting into the nucleus and mRNA coming out of the nucleus to get protein expressed. So this is a very critical protein complex. And this is the computer model for this protein complex. And we've been trying to image them. This is some of the very new data looking into the nuclear pore complex in yeast. So here is the nucleus of the yeast. We can zoom into here, and this will be a nuclear pore complex, and that's we want to look into it. The neighboring efficiency is not 100%, but for those neighbored, because we're supposed to see 8 of them following exactly 8-fold symmetry. We see 9, we see 5 of them, but yes, we can resolve individual nuclear pores, individual proteins in the structure. And this is where we are trying to follow up on to determine the position of each of the nuclear pores in the complex to allow us to reconstruct the structure. This is getting into higher spatial resolution. The other direction is to look deeper into a tissue sample. And to image away from the tissue sample, one natural challenge is when you're trying to use an oil immersion objective to look into water, and there's a refractive index mismatch that can cause a lot of spectral aberration when you are, say, 10 micron or 15 micron away from the surface, not too far to get into the scattering problem yet. And we've found solutions, buffers that can match the refractive index with glass while still make fluorophores fluoresce and photo switching. And another thing is to build an active stabilization system for the microscope. And that doesn't rely on adding fiduciary markers. And it's easy to add fiduciary markers to the glass surface or if you have cell samples. But if you have a big piece of tissue, it's not easy to stick something in the middle of the tissue or make sure that your structure of interest is still right next to the fiduciary marker. So we have a way to do 3D active stabilization without any special treatment to the sample by taking transmitted light image or dark field images. So this is our effort in getting stronger tissue imaging capability. The one that I'm going to spend most of the time today on is how we can image faster. You know, for light microscopy, the biggest advantage, of course, is light cell imaging. I mean, we claim that this is minimally perturbative. It's unlike electron microscopy, which has to fix the sample or freeze it and throw it into the vacuum. Here, we can do things within life. And so one thing that we wanted to follow is the mechanism that controls the growth of dendrites from neurons in the Drosophila embryo. And this is just the picture showing 11 hours, 13 hours, 15 hours after the egg of the fly is hatched. And then you can see at this point, the dendrites start to grow out from the neuron. And we want to image that. And so this is the conventional fluorescent image. And with the tissue imaging, well, some of the effort in creating better, by far not the best, better tissue imaging technique, we can resolve these neurons. We have better image of the projections, and more importantly, at the base of the dendrite growth. So you are looking at the cross section of the neuronal projection. You can see this protein pack is accumulated towards this upper side, and that's where the dendrite is going to grow up. And so this is some of our efforts trying to clarify what's the mechanism that tells at this time, at this point, the dendrite is going to grow out. This is still not quite challenging yet because it takes hours for things to happen, and that's easy. And for some of other things, we really want to image faster, and that's a challenge. So it comes down to live cell storm. Live cell storm, live cell palm, it's not that difficult. And as I said, we're going to come back to the imaging scheme again. We take a huge stack of images. In each stack, a random subset of molecules in the sample is switched to fluorescent state. So we'll capture all these images. For each stack, for each frame in this movie, we determine the molecule positions, and we sum them all together. That's a storm image. But if we don't sum them all together, instead we divide into shorter time steps. For each time step, we create a storm image reconstruction, this will be a movie. For example, this is when we look at a cell with microtubules fused with tubulin in microtubules fused to a photoswitchable fluorescent protein. So that by itself is fluorescent. Basically, this is by far the easiest storm sample. And this is experiment that I did for the prospective graduate students when they visited UCSF. So we ran a demo experiment. So if you do some all together, you do get a storm image of the microtubules. And if you cut into 1,200 steps, 1,200 frame steps, and that you see the microtubules moving. So this is a storm movie. And camera is acquiring at 60 frames per second. So 1,200 frames would correspond to 20-second time resolution. 20-second time resolution is sufficient to see, for example, some of the microtubule dynamics. That's not a problem. But for many of the other similar processes, this is way too slow. Why can't we get faster? Why do we have to use 1,200 frames? Well, this comes to fundamentals of the sampling theory of storm again. Say, we go back to our 40,000 frame microtubule image. If we're not using the whole 40,000 frame, instead we're using, say, 500 frames, we definitely have fewer points. And if we use 200 frames, of 500 frames, 1,000, 5,000, 40,000 frames, you can see with more points, the image looks better. And about 500 frames, you can start resolve individual microtubule very reliably. This is in the case when microtubules are rather sparse. When the microtubules are dense, say in this case, you would need almost 5,000 frames to be able to reliably say that, okay, I can resolve individual microtubules. So there's a connection between the density of sample points and the effective resolution. And a simple explanation is the Nyquist criteria, because when you have two structures, when the sampling density is not enough, you don't resolve it. Only when the point-to-point distance is shorter than 1/2 the feature size you want to resolve, you can see that, okay, I can resolve that precisely. And in fact, this applies to all fluorescence microscopy method, because we all have discrete fluorophores neighboring the sample. So it's very easy to understand for storm, because the final image is made up individual fluorophore points. But in fact, it's applied to all fluorescence microscopy methods, especially when the resolution is high, when the resolution is comparable to the distance between two fluorophores in a sample. And the Nyquist sampling criteria will be a pretty much oversimplification of this story, because it assumes uniform sampling. When we start to consider random sampling, and it's much more complicated. There has been paper coming out recently in PRL about the real resolution of the storm. And so what does this mean? So come back to our live storm imaging. We have our single molecule image. Because we're using red 4 the full width half exponent is about 320 nanometer. And because we want to do single molecule imaging, we don't want two molecule images to overlap. So we have to set them fairly sparse. And supposedly, let's do some very simple calculation. One molecule occupies a 500 by 500 nanometer area. And because the switch is stochastic, if you have on average one molecule in this area, you're going to have 30% of chance of no molecule, 30% chance of one molecule, and 30% chance of more than one molecule. That's Poisson distribution. And if you have more than one molecule, you definitely have overlaps. So usually, to avoid overlap, you have, say, on average, 0.1 molecules in that area. That's 0.25 micrometer square per frame, per micrometer square per frame. With this density, then you can calculate to get a 35 point-to-point distance. That means a 70 nanometer density nanometer resolution. You need 2,000 frames. And if the camera is working at 100 frames per second, this is the kind of rough order of magnitude of the most popular camera that people use on storm microscope. This exactly translates to 20-second time resolution. So if you read all the live cell, most of the live cell storm image, you see that the resolution is around 60, 70 nanometer, and the time resolution is about 20, 30 second time scale. And the caveat here is we're calculating a 2D uniform sample. So if molecules are distributed uniformly. If the molecules are distributed non-uniformly, then we can do faster. But as I said, 20 seconds is by far not sufficient. We can increase the frame rates. And there's a recent paper by Xiaowei's lab who used a 1,000 frame per second camera. And that definitely gets to 2-second time resolution. The problem is not all of fluorophores can switch that fast. For example, fluorescent proteins cannot do. So can we increase this number? Because that's the other limit of our location or our sampling rate. So if we want to increase our sampling rate, that can be a disaster. If we have too many molecules per frame, and there's no way we can do single molecule nucleation. And to solve this problem, it comes to an algorithm which I think many of you are familiar with. It's called compressed sensing. And I'm just listing the title, which I've been very much impressed by how people can formulate a good reading title, like robust uncertainty principles, exact signal reconstruction from highly incomplete frequency information. Yeah, that definitely sounds something. And so this is by Emmanuel Candes and now at Stanford University. And the story, I didn't know compressed sensing before. Why did I come to compressed sensing? Is one day I was at a restaurant in San Francisco, a Chinese restaurant in San Francisco, trying to get some takeout foods. But they got overly popular. I actually literally waited for 40 minutes for them to cook the food. So I was too bored and picked up the phone, called my former roommate at Stanford, who's now at assistant professor at Georgia Tech. And I said, I had this problem. Want to do single molecule nucleation in these images. He thought for about 3 seconds. He said he's got a solution. And that's compressed sensing, because that's what he's been working on for CT reconstruction. And how compressed sensing can be applied in this case? So the imaging procedure of in whatever night microscopy, you have a collection of molecules in your sample that's giving out fluorescence. And that convolves with the Poincare function of your imaging system to get a camera image. With the storm, what you do is you want to go from a camera image back to know where the molecules are. And there's one thing you can use. Because our photo activation scheme, even when the molecules are pretty crowded in the camera image, we still have a sparse collection of molecules in original space. And that sparse collection would allow us to use, that sparse constraint would allow us to use compressed sensing by minimizing the L1 norm, subject to this convolution. Of course, we have to count in the short noise of photon counts. But if you do this optimization, you can get this. So for example, some simulations, if we simulate 10 molecules in this field of view, actually we have to try our random number generator quite a lot of time to create 10 molecules that without any overlap. Then we can use fitting method to pick all 10 of them. And compressed sensing would return a much sharpened image with a pixel size that's one camera pixel divided into 8 by 8 in this new matrix. And that we found out 10 molecules. And if more molecules, 50 molecules, fitting identifies not so efficient. And compressed sensing identifies 45, where fitting identifies 8. We have 100 molecules, then I don't need to show fitting. Compressed sensing still identifies 94. And 200 molecules, that's to the extreme. And then compressed sensing still identifies 156, even when things are very dense. So these are simulations that accounts for the photon variation from molecule to molecule, as well as the short noise, as well as the fluorescence background. Try to match the experimental condition. Overall, if we want to compare fitting methods, that peaks at about 0.5 molecules per micrometer square. That's exactly our assumption when we do the back envelope calculation. And compressed sensing, so this is where 100 molecules goes. This 200 molecules goes 9 molecules, almost 9 molecules per micrometer square that it can identify. So that's more than an order of magnitude improvement in terms of the sampling rate. Of course, there's one caveat. I mean, you pick those molecules. Are you picking it right? But in our simulation, we can compare our result to our simulation input. And the precision of matching the comparison result to the simulation input does get worse and worse when we have a higher density, so which is expected. At 8 micrometer per square molecule density, we are getting about somewhere 90 nanometer full width half maximum in terms of this matching. So that's not too bad at all. If we go even higher density, then that gets close to about 220. That's structural information microscopy resolution. That's why I didn't go even higher in terms of molecule density. So we are able to identify molecules precisely. And some of the real example of imaging fixed microtubules, and that's conventional fluorescence. And in the storm raw data, we have very high activation, so it's completely overlapped. With fitting by 100 frames, not really seeing anything. 500 frames, you can probably see something. By compressed sensing, even 100 frames, you are getting a good enough image to resolve those microtubules. And we can also do live cell imaging. This is the same MEOS2 tubulin S2 cell, actually acquired on the same day. And now instead of 1200 frames, we've been using 169 frames, that's 3 seconds, to reconstruct an image. So that we can see microtubule dynamics with 3-second time resolution. And again, this is the image taken on that day when I was ran demo experiment to the prospective students, and it ends up in the paper. Quite impressive. Yeah. So we can do fast. Can we do it faster? 3 seconds. Yeah, 3 seconds impressive. Can we do even faster? So there's another system that we're trying to study, is the plasma membrane of the cell, and particularly the lipid in the plasma membrane. A lot of the cells, when cells make communications to the outside environment through all the proteins and lipids and signaling molecules on the cell membrane. And there's lots of complicated things going on. And one thing we've been trying to study is the lipid. And for that, we've been using a dye. It's called DID. It's a very common lipid dye. And the DID, interesting thing is the chromophore structure is exactly the same as Cy5. So it is photoswitchable under exactly the same condition. And so we can use DID to stain the cell. That's probably the easiest stain you can get, because it takes 5 seconds to stain the cell. And you can do storm image with that. And this is the conventional fluorescent image. We will turn on laser to do the photo switching. You can see they are blinking and they are moving around. These are small molecules, cannot be fixed, and they just move around very fast. We can still do the nucleation. The thing that they are moving around actually helps us, because they help us sample all the different positions on the plasma memory. So we get this nice image of the morphology of the cell. And we can image a lot of different cells. And we can see some interesting structure in plasma membrane. We haven't tried to figure out what they are, like these. And sometimes we haven't tried to figure out whether they are real or even just some staining artifact. And sometimes we see this hole in the cell, plasma membrane. We don't know what it is. Maybe some region that the idea is excluded. It's possible. It's definitely possible. And so that sounds easy. That's a big problem. As I said, the idea is moving around. It's actually moving around really fast. And if you do the calculation of its diffusion coefficient, 1 micrometer squared per second, in 10 milliseconds, it's going to diffuse for 200 nanometer. That's larger than our pixel. That's definitely you can see it. That you see this single molecule spot, this is not round, this is not round. Many of things get dim and it's just not round because they are moving. And the issue is that we try to increase our frame rate to 100 frames per second. So that's 10 millisecond exposure time. And 10 millisecond exposure time, it's moved in, the mean square displacement is 200 nanometer, and that's making the blurry thing. So the trick we use, instead of increasing the camera frame, we cannot do that on our camera, unfortunately. We need to spend $30,000 to buy another camera. And we just shutter the laser faster, of course, corresponding increasing the laser power. So this way, with strobe laser, shorten the exposure time to 500 microseconds to 1 millisecond. And now you see a much better image because we still keep the same amount of energy in each frame. And this is the key to get good daddy image, to really get the exposure time down to sub-millisecond. And another thing for 3D imaging, especially when you're using astigmatic imaging, you have completely rely on the shape of single molecule spot to determine the Z position. So this is not acceptable. And when we strobe laser, we can do 3D imaging much better. So this is a comparison of a flat sheet of membrane and the distribution of the Z. And when we start to strobe laser, we see a much narrower distribution in our Z position because the shape is more cleaned up. And that we can, in fact, image the 3D topology of the plasma membrane. And again, we haven't yet tried to explain what these structures are, but it will be very interesting to figure out how different membrane proteins is associated with which membrane microdomain. Even more interestingly is try to utilize the fact that they are moving around. So they are switching, they are moving. They can be on for multiple frames, and then we can do single molecule tracking. And in this way, because in fact, a fundamental difference between this way of single molecule tracking versus what people have done before. What people have done before, they have to start with a sparse set of molecules. Once these molecules are bleached, they are gone. So they use quantum dots. They can take extremely long traces, but very few traces. This way, we have our DID that's been tracked. We have a huge reservoir that has 1,000 times more DID molecules there. That's non-fluorescent. That will come back. That will come to fluorescence sometime in the experiment. So we have shorter tracks, but we have enormously high density of all these tracks that covers the entire plasma membrane. So these are the tracks. We can even build a high-resolution diffusion coefficient map of the lipid in the plasma membrane. And hopefully, using this way, we can start to resolve some of the lipid macrodomains and see how that's correlating with protein structures. I mean, in the end, we are having an image method. So we can see how that's correlated with protein structures in a cell. And of course, all this, we definitely need to go to very high temporal resolution in terms of tracking. So that's how we are trying to pursue faster and faster processes. But there's one thing, if you go back and think about, for storm, I mean, what's the fundamental of storm? I mean, what is really about? Storm is basically switching molecules. So even when you have a very high density of molecule to start with, you can do single molecule experiment. It's basically stochastic single molecule sampling. After sampling single molecule, what you can do? You can determine the positions. That gives you the imaging method. That's the storm, PALM, and all the D-storm, Palmyra, and GISTM, and yeah, all these different methods. You can do single molecule tracking. You can do many other single molecule things. You can do single molecule anisotropy measurement. This is being done by Sam Hess group. You can do single molecule fret measurement. And that hasn't been turning into imaging method yet, but has been used to distinguish a complicated thread structure to dig a single molecule thread out of that. And of course, it's a general method to take single molecule images when you have a very high starting density. So you can, for example, to do single molecule imaging under physiological concentration. And there's a recent paper by Antoine van Orgen and utilizing this way to do single molecule tracking on flow stretch DNAs. So this, I would say, is the real essence of STORM. And for us, definitely our dream is that if we can do single molecule study in living cells, if we can know how every single molecule is working in a living cell in real time, of course, many of the most interesting questions in biology will be answered rather easily. This is still so far only existing computer animation or in our imagination. We're trying to get close to this stream. And so that's the group of our people, the single molecule dreaming team or daydreaming team. And this is last year and now is this year, so the group has expanded a lot. And also shifting from doing science to having fun. And this is all of group members. It's a big collaborative effort of biologists and chemists and physicists. And also thanks a lot to many of the collaborators that we have at UCSF and other universities, and of course, people giving money. And one final thing I want to show. So I was something at the beginning. I was saying we need to design a logo for the lab, so that we can show it on the group homepage. And the logo needs to contain something saying that we are dealing with light. And then Robert Casper, who's the postdoc here, said, "No, we don't need to do that. It's already there. It's already in the seal of University of California. Let it be light." That's why we're here. Thank you. Questions?
I was wondering, in the reconstructed image, when you see heterogeneity, how do you decide if it's due to actual structural features or just the randomness of the labeling or the randomness of the ones that you activated? I guess it would help if you have some idea of what the structure should look like, but when you don't really have a very clear idea.
That's a very, very, very good point. One thing to say is anything at single molecule level is stochastic. At single molecule level, you can see anything if you want to observe. So there's no point of it makes no sense to interpret single molecule data without statistics. And so for any interpretation of STORM image, is a very much underexplored area, but it definitely relies on statistical tests. And that will allow you to see whether this is real fluctuation or under your experimental condition, under your sampling rate, under your localization precision, there's no way I can tell whether it's real fluctuation or from experimental fluctuation. Yeah. And there are people working on that. For example, because analyzing this type of image, there hasn't been many effort into this because these images not made up pixels. They made up these are scatter plots. So it's this collection of coordinates. How you can calculate the Fourier transform on it? How you can calculate correlation function on it? How you can calculate other things on it? It's not a very well developed method. Recently, there has been paper from Jennifer Lincoln-Schwartz group calculating correlation functions of STORM and PALM images. And there has been some effort of a Stanford group, Merkel, and Mark Schneiser, I think. Yeah, I'm very bad at remembering names. Try to define yeah, the information content of storm image and the information transfer function of this imaging process. I think all these efforts is extremely essential to correct interpretation of storm images. Yeah.
So when you reconstruct the image, do you use another imaging technique to cross-check the results, or do you just leave the instructions what it is?
Okay, that's another point, validation. In many cases, there's no other method that you can cross-check. Because this is the first time you see this. There are some cases, for example, microtubule. There are plenty of studies of the microtubule structure itself. So you can compare what you get with what people initially has characterized using electron microscopy about the microtubule structure. If you have things on DNA, you know how many base pair away they are, you know what distance supposedly you have. So there are cases where you have references in other things. But in many other cases, when you're trying to explore into unknowns, it's difficult. It's the same problem for many of the structural biology methods, like crystallography, like single particle cryo-EM. When you really read more into that, you'll be scared by how easily these supposedly very physical methods can subject to artifacts. And validation is again a big deal in those things. Often, for example, you can try. They try to split the data into two halves and try to make reconstruction to see, okay, if they look the same. But in general, it is a challenge. And with that said, it means, need to be very careful about interpreting the image and also the biological controls. For example, if your structure suggests that this thing has this function, then you have functional study like knocking out, like overexpression, like domain mutations, all these biological controls to see if they are consistent with your imaging result.
Yeah, on one of your slides, you mentioned that you were trying to improve axial resolution by putting a mirror behind the sample to look at the imaging pattern. I was wondering if you could explain a little more how that might work.
So, okay. The methods, the principle is published by, I think 2 years ago. Yeah, almost 2 years ago by a French group. It's so-called time reversal. And essentially you have a phase modulation pattern so that you can focus the laser at two focal points. One in front of the mirror, one behind the mirror. And then the mirror, in fact, will fold it back to the same position. And then you have a reverse the phase pattern. And that will then be able to convert the direct instance versus reflected instance into a plane wave so that essentially the molecule and its mirror image, one is in front of the camera, one is behind the camera. That will make them merge at the camera plane so that they interfere. It sounds simple, but we have been running into big problems. For example, we cannot image too far away from the mirror because the coherent length of fluorescence. So these are real limitations for this method. And we have had very interesting discussions on this. Yeah.
Can you comment on the reconstruction time using compressive sensing?
It's very, very, very slow. So we actually wrote in the paper, I regret that we put in supporting information. I should have put it in the main text saying that it is slow. But the algorithm we're using, we're dividing the image into small patches and run optimization on each patch completely independently. In fact, make it very available to massive parallel computation like GPU computation. So supposedly, if we migrate all the code into GPU, it can be orders of magnitude faster because we do optimization on 7x7 small patches completely independently. So you can easily divide everything up and do the optimization. Or even if we're not doing that, each frame is independent, so that we can also do parallel computation. Yeah.
So I'm interested in the storm videos you were showing, and it looked like there you kind of see microtubules with bright areas moving around on them. So when you're looking
What those are? Yeah.
when you're looking at the labels moving, so what does that actually tell you about what the cell is, or the microtubule?
So that, yes, this is one thing, that fluorescent protein, although supposedly it comes up and then get fully bleached, but they blink a lot, and that has a artifact that has only recently caught people's attention. People initially thought we can easily use fluorescent proteins to count absolute copy number, but we've tried, very difficult to correct for overcounting and undercounting. And so, yes, you will see fluorescent protein moving on non-microtubule, but also you can, if you see microtubule move naturally, that will be the microtubule movement itself. One thing is, with faster acquisition, faster means we definitely want faster switching fluorescent proteins. So in the end, if you're talking about making better fluorescent protein for live cell imaging, this is the direction. We want fluorescent proteins that switch fast and can switch for many cycles, because we still want to observe it multiple times. So far, there's none that can do this. Yeah. There are reversibly switching proteins. There are fluorescent proteins that can switch for 1,000 cycles, but they are just not perfect yet.
If there are no more questions, let's thank Professor Huang.
Thank you.